Hi students, welcome to my channel Easy Learning. So today I will be dealing with the topic asexual reproduction. In the previous video I have explained about sexual reproduction. The link is given in the description box, and the first chapter link is also given there in the description box. Life process, all the 4 life processes. Okay, so today we will deal with asexual reproduction. So first I will move on to the definition of asexual reproduction. Already I have explained regarding the definition, even though once again I will tell that asexual reproduction means the method of reproduction or the mode of reproduction in which the new organism is produced from the single parent without the involvement of the sex cells. That is known as asexual reproduction. So under asexual reproduction, there are 6 methods for you to study. The 6 methods, the first one it is fission. Second one it is budding. Third one it is regeneration. Fourth one it is fragmentation. Fifth one it is spore formation. And the last one it is vegetative propagation. So these are the 6 methods of asexual reproduction. First one it is fission, second one budding, third one regeneration, fourth one fragmentation, fifth one spore formation, and sixth one it is vegetative propagation. You should know the definitions of all the 6 methods, and among that some of the important areas I will be just pointing out so that it will be helpful for you for board exam. The board exams are there for you to concentrate in those areas. Only the important areas you have to concentrate so that it will be helpful for you to score maximum marks, that is 100 marks in the subject science. Okay, so now we will move on to the first asexual method, fission. So what is meant by fission? From the term itself we know it is splitting. Fission means splitting. So the splitting up of the parent organism in order to get the new organism. So here the parent cell, this fission process, it is mainly taking place in the unicellular organism, that is the protozoans, that is amoeba, then leishmania, paramecium, etc. So this fission process, it is taking place in the unicellular organism like protozoan, that is amoeba, leishmania and paramecium. So the splitting up of the parent organism to get new organism, that is known as fission. So this fission is of 2 types. One it is the binary fission, and the second one it is the multiple fission. So it is bifurcated, or it is classified into 2. One it is the binary fission and the other one it is the multiple fission. So first we will move on to binary fission. Binary fission means the parent organism splits into 2. Organism just break or split into 2 new organisms. That is meant by binary fission. And second one it is multiple fission. That is from a single cell, large number of new organisms are produced. That is meant by multiple fission. Multiple fission means the parent organism splits or breaks into large new organism. So the single cell it is broken down into many new organisms. That is meant by multiple fission. So you should know the definition between the binary fission and the multiple fission. So this binary fission, it takes place in amoeba, leishmania and paramecium. So these are the 3 examples for you to study. I am not going in detail. In that, the important part I will be focusing. That is, in the case of binary fission, you should know the diagram of binary fission in amoeba. A very few steps I will tell regarding the binary fission in amoeba. Once the amoeba attains its maximum size or the growth, then first the nucleus will split or divide, and then into 2, and after that the each nucleus is surrounded by the cytoplasm and then the two new organisms, that is daughter amoebae we will tell. So, the two new organisms will be formed from the parent cell. So, after the daughters are formed, then the parent organism does not exist because the parent organism is only splitting or two new organisms. Okay, so that is regarding binary fission. And the same way the process takes place in paramecium also, and in the case of leishmania. Leishmania, it is a parasite which produces the disease known as Kala Asar. That you have to remember because it comes for MCQ. It is a parasite and it causes the disease known as Kala Asar, leishmania. The diagram of leishmania was given and it was asked to identify the particular organism. So, the leishmania, the structure it is this type of structure with a flagella. This is a flagella, it is a locomotory organ. So, here the leishmania, the division takes place in the longitudinal way, that is through the flagella, it gets divided and identically two halves it will be forming. That is how two new organisms will be formed in leishmania. So, it takes place longitudinally in a definite plane. The division it takes place in a definite plane. Here one question comes, give reason question. How the division or the binary fission in leishmania is different from that of the binary fission in amoeba. So, it is a frequently asked question. So, you have to concentrate in that particular area. Here in leishmania, there is a definite plane. Through the definite plane only the division takes place. Whereas in amoeba, in any plane, you can divide. Amoeba, it does not have any particular structures in it. So, you can divide in any way. You can divide in any way to get two equal halves. So, in amoeba, the division takes place in any plane, whereas in leishmania, it takes place in a definite plane. So, these are the main difference between the binary fission in which takes place in leishmania and amoeba. So, it is a frequently asked question. You have to concentrate in that particular part. Okay. Next is regarding multiple fission. Multiple fission means the organism. It splits the single cell. It gets splitted into the parent organism. It gets divided into many new organisms. So, here that under unfavorable condition, that particular cell, it is protected. How this is the cell means, then it is protected by a cyst. A cyst will be formed or a protective coat will be formed in the unfavorable condition. So, at that time, the nucleus which is present there in the cell, it goes on dividing. And then each nucleus, it will be surrounded by some amount of cytoplasm and a thin membrane will be formed. Like that, many new cells will be present there inside the cells. So, once the favorable condition occurs, that is the temperature and warmth and all it is available, then the nutrients and water, everything it is available, then the cyst will break open. Like this, the cyst will break open and new organism, that is the new cells will be formed. It will get dispersed out. Okay, that is how the process of multiple fission takes place in single cellular organism. This is how the single cell parent organism, it gives rise to many new organisms by the process of multiple fission. So, simple example for multiple fission, it is the plasmodium. It is a malarial parasite which causes the disease malaria. So, plasmodium is the simple example for multiple fission. So, that is all what you have to concentrate under the topic fission. Next one, it is budding. In that, you should know the term what is meant by bud. Bud means it is a small projection on the body surface. That is a small outgrowth which is formed on the surface of the body. It is known as bud. So, that budding, it takes place in two organisms. One, it is the hydra and the other one, it is the yeast cell. These are the two examples you have to study under budding. So, bud, it is a small projection that comes out from the body of the organism. It is known as bud and that process it is known as budding. So here also in the case of hydra and yeast, the diagram of hydra and yeast is very, very important. The budding in hydra and budding in yeast, the diagram is very, very important. Here the binary fission, the diagram of amoeba, it is very, very important. Okay, so diagram you have to concentrate. So in the case of hydra also, the hydra, a small projection, it arises from the body of the parent hydra. Then after it has attained some size, then the tentacles and the mouth will arises, and that tentacles and mouth, once it is arised, then it gets detached from the parent organism and then it grow into a new organism. So in a simple way I have explained the budding in hydra. So that is how the process takes place. Same way in the case of yeast cell, yeast is also a non-green plant, it is a fungus. So the yeast cells, a small projection arises. This is the yeast cells, means a small projection arises in the yeast cell. Then the nucleus get divided to the projection also to the bud, and then afterwards, once it has attained its size, it gets detached from the parent organism and then it grows into a new organism. But in the case of yeast, the division it is taking place in a much faster rate, that before it gets detached from the parent organism, its outgrowth arises. Like that, a chain of yeast cells will be formed in the case of yeast. And afterwards, it gets detached from the parent organism and it grows into a new organism. That is how the process of budding, it takes place in yeast cell. I hope you all have understood regarding fission and budding. Next, we move on to regeneration. Regeneration is the third asexual reproductive method. So, from the term itself, we know that regenerating, it is again forming, regenerating. So, the definition of regeneration, it is the forming of new organism from the cut body part. If the body part of an organism is cut, and that cut part again it will regenerate or it will form into a new organism. That process it is known as regeneration. Here you have to study the example for planaria in regeneration. Planaria is the example which you have to study for regeneration. So, on its own, naturally only some cuts may occur on the body of that organism. It may be cut into 2 or 3 or many pieces, and each piece will grow into a full organism. Okay, so that process it is known as regeneration. The example it is planaria. So how this process takes place? That is, this method of asexual reproduction, it undergoes the process of growth and development. It undergoes the process of growth and development. Due to growth and development only the process of regeneration takes place. So here the particular body part of an organism, if it has been cut, that cut body part, it undergoes the division of the cell, and then cells get divided and then a ball of cells will be formed. Then afterwards what happens? In that ball of the cells only, the cells moves into the particular areas to form the various organs. Then that particular tissue, that is the group of the cells, it will change its shape to form the tissues, and the tissues then it will form the organ, and then the organism it is formed. That is how the process takes place in the regeneration. So the main process under the asexual method of regeneration, it is growth and development. First it grows, that is the division of the cells goes on. Then once the cell is divided, and then it will move into this particular place, and then it forms the tissues, and then it changes its shape to form the organ, and then it forms the organism. So that is how the process of regeneration takes place. Here one important question is there. Justify the statement or comment on this or give reason type. That is, regeneration process is insufficient for the large multicellular organism, or why the regeneration process does not take place in large multicellular organism. So the reason it is that in large multicellular organism, the organization of the body is very complex way. It is a complex process. It is not a simple organization of the body. But in the case of these organisms, the organization of the body is very simple, so that only very less specialized cells are present there in these organisms, which help the cut body parts to grow into a new full organism. But whereas in the case of multicellular organism, their body organization is very complex, and large amount of specialized cells will be there, and those specialized cells will form the tissues. tissues will form the organ will form the organ system, and then it forms the organism. So it is example which is given there in the book. It is regarding in certain dogs, their tails are cut, and that cut tail will not grow again into a full dog. So that is the simple example which is given. So sometimes the question may come with example, you have to justify the statement. Then you can explain like this, because the body of multicellular organism, the organization is very complex. They have got very large number of specialized cells. But whereas these organisms, they have got only very less specialized cells, and it is easy for that cut body parts to grow into a full new organism. So that is regarding regeneration. The next method, it is fragmentation. So regarding fragmentation means the method of asexual reproduction in which the matured organism, it breaks into many fragments, and each fragment it will grow into a new organism. The simple example for fragmentation, it is Spirogyra, an algae plant which is found in water, and on maturing, on its own, naturally, nobody is going and cutting it. So on its own, naturally, each fragment it breaks, and then each fragment it grow into a new organism. That is how the process of fragmentation takes place. The fifth method, it is the spore formation. Microscopic asexual reproductive bodies which is present in the atmosphere, that spores are present there. So once it gets favorable condition, then each spore will grow into a new organism or a new individual. So here the spore formation, it mainly takes place in Rhizopus. This is the scientific name. The common name it is bread mold. The common name it is the bread mold. So Rhizopus. The spore formation, it takes place in Rhizopus, and other than that, non-flowering plants like ferns and mosses, then mucor, all these undergoes this particular asexual method known as the spore formation. So the spore formation, the spores will be present in a spore case or the sporangia under unfavorable condition. So once the favorable condition occurs, then it bursts open and the spores will be dispersed into the air. And it will be, after once it is getting dispersed in the air, then it will fall off either on the food or any other surface soil, and then it grows in that particular area. Okay, that is meant by spore formation. So here the simple example it is Rhizopus, that is the bread mold. So simple in earlier classes, in lower classes like 6th, 7th and all, you might have done that particular activity. You take a piece of bread and then put moist it, and then you put it in a box. Okay, keep it for 3 to 4 days. After 4 days, we can observe that a blackish spot or a greenish spot will be present there on the bread piece, isn't it? So that is the bread mold or the Rhizopus which has grown on the piece of the bread. Its root, it is known as the hyphae. When it has got a thin stem and then a bulge-like structure, it is known as the sporangium, in which the spores are present. This is the structure of the Rhizopus or the bread mold. Okay, so that is how the process of spore formation or this asexual reproductive method, it takes place in these organisms. Last method, it is vegetative propagation. Vegetative propagation means from the vegetative parts of the plant, you are getting a new plant. That is meant by vegetative propagation. Vegetative part means either it can be root, it can be stem, it can be leaves or buds. So from the vegetative part, you are getting new plants. That is meant by vegetative propagation. In that, we are studying here mainly based on artificial propagation. So vegetative propagation, you will be studying under, for example, in the case of leaves. The leaves, the buds are present there in the leaves, and from each bud, the new plant which arises once it gets the favorable condition like nutrients, water and temperature. Isn't it? Same way, in the case of a simple example for leaves, it is the bryophyllum. Then in the case of the stem, you can give many examples regarding hibiscus or rose or whatever it is. And then regarding based on the root, that is the underground stem or the modified root, we can tell. In the case of potato, the small scars which is present on the potato, you can just remove it and then you can put it in the soil or any other condition where the water, temperature and nutrients are available. In that particular condition also it can be kept. From each scar, it arises a new plant. So, that is in the from the vegetative part you are getting a new plant. So, mainly based on this artificial propagation, when we are talking about artificial propagation, under that you have to study three main methods. The artificial propagation, you have to study three main methods. First one, it is cutting. Second one, it is layering. And third one, it is grafting. So, cutting, layering and grafting. These are the artificial propagation methods. Why it is known as artificial propagation methods means, otherwise it is known as man-made methods. The small part of the plant, it is cut with the help of a knife and then it grows into a new plant. For example, simple example, if you are cutting the stem of a hibiscus or a rose plant, that cut part, that is the stem part of that plant is buried in the soil and then it is provided with all the nutrients, water and temperature, everything, and then it is growing into a new plant. So, that is, you are getting large number of plants by man-made methods. That is why it is known as artificial propagation. Okay. So, here in the case of layering, under cutting already I have told. The second one, it is the layering. Layering means the one branch of the tree, it is being bent and then it is buried in the soil. It is covered with the moist soil, the part which is touching the ground, and that area it will be covered with moist soil, and then the other end of that particular branch, it will be exposed over above the ground. And once the root arises, where the part of that particular branch which is buried in the soil, once the root arises, then it will be cut from the parent plant, and that part of that branch, it grows into a new plant. That is meant by layering. So, the diagram is, if this is the tree and one branch is there, this branch, it will be bent in such a way that it is touching the ground, and then the other part, it will be exposed on the, so this area, it will be covered with soil. So, after giving the sufficient amount of water, nutrients and all, what happens? The root arises from that part which is buried inside the soil. And once the root arises, it will be removed from the parent plant, then it grows into a new plant. That is the method of layering. All these, it is very important. You should know the definitions, what is cutting, what is layering and what is grafting. Grafting is more important. You should know in that two terms, what is stalk and scion also. So, here the third method of artificial propagation, it is grafting. So, grafting means the process of artificial vegetative propagation in which the two plants, that is the cut stem of two plants, it is attached or joined, and then it will be grow into a new plant. For example, the plant which is having the root, that is we are cutting a plant and this part, that is which is having the root. So, when we are cutting, we will cutting in the plant in a slanted manner, isn't it? So, this part, it is known as the stalk, and another stem of the plant which we are cutting, it will be attached to this. It will be attached to the stalk. So, this it is known as the scion. So, the part of the stem which is having the root, it is known as the stalk, and the part of the stem which is attached to the stalk, it is known as the scion. So, when you are attaching, you are keeping in such a manner that the cambium of both the plant, it get attached together, because cambium helps in the growth of the plant. Okay. So, the cambium should be attached together, that is both of the scion and the stalk, and then it is covered by the cloth, and then it will be covered with the plastic, so that the bacterial infection does not take place, and the water also will not be lost from this both scion and the stalk. Okay. So, that the infection does not take place with the help of the microorganism, and the plant should not get dried off. So, for that purpose, we should be careful while doing this grafting process. Okay. So, the two parts, one it is the stalk and the other one it is the scion. So, two parts of the plant. Okay. So, here you should know regarding the definition of cutting, definition of layering and grafting. It is very, very important, the artificial vegetative propagation method. Okay, cutting, layering and grafting. It is always asked. It is a 3-mark question, repeatedly asked. So, you have to concentrate more in this area. Okay. Next, regarding the advantages and of artificial propagation and advantages of grafting, it also comes for exam. You have to know regarding what is the main advantage of grafting and main advantage of artificial propagation. Next topic, which comes under vegetative propagation, it is the tissue culture. Tissue culture. Tissue culture means it is a method by which we are getting many new plants from the cells. Okay, from a group of cells. So we are cutting the topmost part of the stem, because that tissue, it is known as the meristematic tissue, because meristematic tissue helps in the growth of the plant. So that meristematic tissue, it will be cut from the tip of the stem, and that tissue, it will be put in a medium, culture medium. That medium, it is known as the agar solution, and it will be providing the needed hormones, the needed nutrients, everything it will be provided. First, the tissues, it will be known as the callus. That is, the group of cells, it is known as the callus. Then it will be put in a solution where the hormones, it will be provided for the growth of the root. Then after that, that particular callus, it will be taken and put in a medium which contains the hormones for developing the shoot, and then it will be separated from that. From that, each plant or sapling, it will be separated and put in a separate medium for the growth of the plant. So the main advantage, it is that disease-free plants we will be able to get. It is only very less space is needed. Then it is a very faster technique, and any number of plants can be produced in any time. That is the main advantages of tissue culture. So tissue culture means from the cells you are getting the new plants. Okay, so that is meant by tissue culture. So now we will move on to the last very important topic. Here the tissue culture advantages is very important. Always the question is repeated for the board exam. So you should know the definition and the advantages of the tissue culture. So next we will move on to the last topic of this particular part under asexual reproduction, whether the organism of the asexual reproduction, they are considered to be the exact copies of the parent. So all of a sudden, if you observe, we can know that they are the exact copies. There is no change in them. But even though during the DNA copying, which takes place by some biochemical reactions, there may be slight variations also can occur in the asexual reproduction also, because no reaction, we cannot tell that it is 100% the same result it may give. So when the DNA is being copied or DNA replication when it is taking place, then they may get slight variation. That is, the exact same DNA will not be formed in the offspring also. So slight variations may occur due to that particular biochemical reaction. So due to that variation, what happens? There may be slight changes from the parent plant and the new plant which it has been produced. So that is why we cannot tell that they are the exact copies. Even though in a physical move, we can tell that they are the exact copies. It will not be easy to identify what is the difference between the parent plant and the new plant which has been formed. There are slight variations. So what is the main significance of the variation? That is very, very important. 3-mark question it is, based on the significance of variation. Variation helps the organism to survive in any drastic conditions. Whatever changes takes place in the environment, if the variations are present there, then it may help that particular organism to survive in that particular new condition which has formed in the environment. Simple example I will tell regarding bacteria. If the new organism which is being formed, if it is having slight variation, so if the temperature of that particular water where they are surviving, if it has changed, then the parent organism cannot survive in that particular condition. But the new organism or the offspring which is produced, since they have variation, they have the capacity to withstand that change which has occurred in the temperature of the water. So it will survive. But if the variation is not there in the organism, then the whole group of organism will get extincted. So that is how it helps. The variation in the organism, it helps to survive in any drastic condition which takes place in the environment. So variation helps in the survival of the organism. So I hope you all have understood. Regarding one more point based on sexual reproduction, it is that why the amount of DNA does not get doubled in the sexual reproduction. That is a very important. Give reason or justify the statement question. It is in the case of sexual reproduction. Here I was discussing about asexual reproduction, but in the type of this type of question, one more question may arise in the case of sexual reproduction. That is why I am explaining it here. That is, the amount of DNA does not get doubled in the offspring in the case of sexual reproduction, because sexual reproduction takes place with the help of the fusion of the male gamete and the female gamete. So this male gamete and the female gamete will be having only half the number of chromosomes each. The sperms will be having only 23 chromosomes and the ova will be having only 23 chromosomes. So when the sperm and the ova, when they combine together, then these chromosomes combine together and then they will be having the total number of 46 chromosomes in the offspring. So since they are having only half the number of chromosomes, the DNA does not get doubled during the sexual reproduction. Okay, so I hope all have understood this particular topic which I have covered today, that is regarding asexual reproduction. We have discussed all the 6 methods of asexual reproduction, its explanation, its definition, the different examples also I have told. So you have to concentrate in all these areas, the diagrams especially, the budding in hydra and yeast and the binary fission in amoeba. It is very, very important. So here you have to concentrate in all these areas wherever I have discussed today. Then regarding the importance of variation, the significance of variation, then regarding the advantages of tissue culture, advantages of grafting, advantages of vegetative propagation. So all these areas you have to cover. Only very short time is there. So in that particular time you have to concentrate only in the important areas so that it will be helpful for you to score 100 marks in the particular subject. So this asexual reproduction, I hope all have understood. The previous all the topics I have uploaded in my video. So to get more videos of the remaining chapters, do subscribe my channel. Okay, thank you children.